So welcome to the session 8, Curation at Scale in the Time of Pandemics. I think the last 2 sessions were actually a wonderful lead into this. And I want to have a discussion with our panelists on the 4 S's: speed, scale, sharing, and standards, and really how the SARS-CoV-2 pandemic has influenced the ability to do curation at scale. So this is a panel discussion with Theo Bloom, Susanna Sansone, Andrew Su, and Rodney Brewster. And I will be moderating the discussion. Just a couple slides of introduction. So how do speed, scale, sharing, and standards interact? Because it's kind of a trade-off. You can increase the speed and scale by faster and wider dissemination of information. For example, we've heard a number of talks on preprint servers. We can introduce new tools to automate parts of the curation pipeline. We've heard a lot about that. And by sharing the curation activity across a broader group of scientists. But all this involves trade-offs with respect to quality. So there's the question of how to balance the rapid dissemination and access to new findings against the need to review, to do expert curation, and to do quality control. So I think this is a very interesting and rich topic. I also, if I can advance my slides, want to make sure we talk about the role of standards. And we actually heard a lot of very interesting talks just now. Standards are, of course, key to the successful sharing and archiving of information, and the absence of standards and standard nomenclatures create obstacles to the sharing of information, to the retrieving of information, and even to the curating of information. So how can standards and expert curation scale up to keep pace with the dizzying rate of new knowledge? And so I hope that our panelists will address these questions. So here, what we're going to do is I'm going to ask each of the panelists in turn, starting with Theo, to introduce themselves quickly. And once that's done, I have some starter questions. They're kind of by topic area, so roughly pre-publication, automated tools and metadata, scalable, sharing the workload, and standards. And I'm going to let each of those run about 10 minutes, but I would encourage the panelists to interact, but also I will try to monitor the Q&A panel and also the chat panel to take questions from the audience. So thank you very much, and I'm going to turn it over now to Theo to start us off.
Thanks very much, Lynette. I hope you can hear me okay. Thanks very much for inviting me to be here. I'm by day the executive editor of the BMJ, the British Medical Journal, but I'm speaking to you today because I'm also a co-founder of the medRxiv preprint server, which was launched in 2019 as a partnership between BMJ, Cold Spring Harbor Laboratory, and Yale. And some of you will have heard from my medRxiv colleague, Richard Sever from Cold Spring Harbor on Monday. And medRxiv was launched with the aim of sharing clinical research results rapidly as preprints. But we were very clear that our aim was to avoid causing harm to individual patients or to whole populations by posting incorrect information or information that couldn't be trusted. So we have slightly more screening than our sibling bioRxiv, but we still aim to post preprints within a couple of days of submission. And at the start of 2020, our 6-month-old server was receiving about 60 submissions a week. And then, as you've been hearing in other talks, COVID happened and getting information rapidly became very urgent. Our submissions doubled every month through the spring of 2020 until we were receiving more than 100 submissions a day in May and June. So we had to very rapidly scale up our screening and evolve our processes as we went along. And quite a lot of what we do relies on authors' declarations. They tell us that they had ethical approval for the work, they pre-registered their clinical trials, they make a statement about data availability. And as a preprint server rather than a journal, we don't check up on what they tell us, and we don't copy edit their text or its metadata to provide links and make sure it's all optimally useful. I sort of feel I should apologize to this crowd for that. The other thing we don't do is use much in the way of AI or machine learning in our pre-screening of articles. We've seen demos of various types of tools, but what we've seen can never actually replace what a human reader picks up by way of concerns about an article. We do use plagiarism screening tools like journal publishers, mainly to check preprints haven't already been posted or published elsewhere. But on the whole, we have a fairly analog system of screening and pushing out the author's text, figures, and associated files so that it's out there and can be mined and used. I'll just finish this intro by saying that as of a day or two ago, I saw that we'd posted 30,000 preprints in our nearly 3 years. A little over half of those are on COVID. And we know we help get information into the hands of clinicians and patients rapidly, but we almost certainly didn't do a good job of getting data into the hands of curators. We rather deliberately leave to journals the tasks of quality control and quality assurance. Thanks.
Thank you. Fascinating. Next, Susanna. You need to unmute.
Thank you very much. Sorry, too many windows in front of me. Thank you, Lynette, for this opportunity. Okay, so Susanna Assunta Sansone from University of Oxford. I am a biologist by training, and I moved in research data management around 21 years ago when I joined the European Bioinformatics Institute to improve data management, data curation, data reuse. So the last 12 years instead, I've worked here at Oxford, where I'm leading a group called Data Readiness Group, which I established. And I'm professor of data readiness in the Department of Engineering Science at university. So our work as a team, it's really in and around improving research practice. So we don't analyze the data, we don't produce data in the true sense, but somehow we do. But we really focus on research data management, on really data sharing, data publication, fairness in general. And I've been one of the FAIR principle, author of the FAIR principle. We work with a lot of grassroots initiatives in the life sciences that develop and improve tools and standards, all the tools and the standards we have, many speakers have talked around these days, which are essential to harmonize the description and the sharing of the data, and obviously are essential to curate the data, to make it reusable, open in terms of format, and maximize the reproducibility as well. We do a lot of co-creation of interoperable data pipelines for other researchers. And we build this educational resource, which are curated resource. So we do curation in a way as well about information that helps other curator. And Alison Lister presented on day one for sharing, which is one of our resource. I love metadata standards, which are the tool of the curation trade, I will say. So, and metadata to me, it's really important because metadata make data count. And so curation make data count. And the FAIR principle, which, as you know, have become, you know, this de facto norm, de facto standard in a way, are really a norm for good data, good research data management, and have aligned all the stakeholders, the funder, the publisher, the library, the curator as well, behind this common set of guidelines. But more importantly, FAIR, it's 80% about good, richly described metadata using community standards. And I will expand a little bit later on, you know, the tool around the community standard. But I think what's really very important that this FAIR ecosystem has really brought renewed energy and attention to data curation in particular.
Okay, thank you, Susanna. Next, Andrew.
Great, thanks. So my name is Andrew Su. I'm a professor at the Scripps Research Institute in San Diego here. My lab really focuses on bioinformatics broadly, and I think in contrast to Susanna's introduction, right? I mean, I think we do try to span sort of that interface between actually doing data mining to come up with testable hypotheses, and also building some tools and infrastructure that may be more broadly useful by the community. So some of the activities that we are very active in include things like building biomedical APIs to give people programmatic access to information. So I talked yesterday about Wikidata and data integration through Wikidata. In addition, we have APIs like mygene.info and myvariant.info, and these are APIs constructed with my colleague and fellow faculty member, Chunli Wu, where we do data integration to try to provide access to integrated data sets in computationally available formats. We are also interested in early on, we had some effort in SARS-CoV-2 research through the development of a resource called outbreak.info. That's the URL, outbreak.info, where we tried to track things like epidemiological information and availability of resources and things like that. This was constructed in collaboration with Chunli Wu, as well as another colleague, Laura Hughes, at Scripps. So in the context of doing outbreak.info, I think there we discovered a lot, and we touched on a lot of the standards that Susanna has been involved in developing, and other people in the community have been involved in developing. So that's definitely an important part of us being sort of consumers of those data standards.
Thank you, Andrew. And Rodney.
Hi, I'm Rodney Brister. I'm the Chief of Viral Resources at the National Center for Biotechnology Information at the National Library of Medicine. I'm also the coordinator for a number of our cloud activities. So we're interested in sequence data and the metadata that accompanies that sequence data. And to be quite frank, we've kind of gone through the fire drill of pandemics before, or the cusp of pandemics before. I actually look back at the Ebola outbreak of 2014 as sort of the touchstone for this current outbreak in a lot of ways. I was actually going to comment on how we reacted. So this is not a model for success. This is simply what we did based on our experience at the time, and probably should be re-examined, right? And early on, and I'm talking December of 2019, of course, we started hearing through colleagues that there was a new virus brewing in China. And we started preparing even at that time. And by February, when the US government began talking about this virus, we were already sort of doing the sort of key initial efforts, I think, that kind of had become our playbook for dealing with pandemics, or not really dealing with, but creating the right sort of archival infrastructure to support data generated from pandemics. And just to say it out loud, our business model is data creators send us their data, and we package it in ways that make it more useful for the scientific community. And so our playbook is first and foremost to make sure that the data creators have the reagents they need. And so our early outreach efforts included some work by Neda Hatcher, where she basically began analyzing a lot of coronavirus genomes and then reaching out to the coronavirus scientific community for help in annotating a RefSeq for what would become SARS-CoV-2. And then some other members of the team began interacting with the ICTV and encouraging them to move very quickly with the classification of this virus and for coming up with a name that could be used across the scientific community. Many will not remember this, but there were actually a number of names for this virus early on. And those two little bits of information allowed people to start talking about this virus, both in an evolutionary sense, the trees that were the early coronavirus trees that were being built, and also in terms of sort of an annotation sense, like this is the best annotation from the community, and sort of set up that next step, which is supporting people submitting data, right? So they have the reagents they need. The second things we did were assessing how big was this going to be. And by February, March, it looked like it was going to be big. My spidey sense was going off and tingling quite a bit. So we had a discussion about how much effort we were going to put into this. And that discussion resulted in Eric's work that he showed earlier. And we went full force on an annotation tool, which is always a risk because we have definitely built them and the sequences never came. But we went full force there to support the sequence submissions. And at that point, we felt we had sort of solved one part of our business model, which was we were providing the necessary tools and reagents to support the actual influx of data. The next steps were pretty ad hoc. It was really talking to the folks that were doing the informatics and asking them what's missing. People like me have a running list of things that need to be improved in terms of metadata, data quality, and whatever. And often it's identifying the use cases that really matter to the community and finding the coalition of the willing to solve the problems around those use cases. And for SARS-CoV-2, there were a lot of great partners at CDC and elsewhere who made this possible. And then we began to sort of move through the list of problems we could solve and working with the community at times to solve them.
Thank you. Fascinating. Okay, I'm going to now go to my list of starter questions. And as I said, you should also the audience should please type questions into the Q&A. But the first question, which I'll let Theo start off with, is, has the availability of pre-publication materials affected the curation pipeline? And you may have sort of answered this, but I'd also be very interested to hear, other people who've talked about this problem weigh in. So Theo, over to you.
Sure. I mean, I think it's interesting because actually sequence deposition is one of those areas where we've had standards and community norms for decades now, such that even preprints, everyone would deposit their sequence at the same time. At one point, we were getting complaints from one of the viral databases that we weren't enforcing particular standards and citation of the database, and we just said, that's not our job. But sequences were being deposited. But I think other types, more complex types of data that require, human intervention to have them adequately curated, it just wasn't happening around preprints, and maybe that part was not being speeded up, but was happening as usual at the time of journal publication. And I guess I should say journals also speeded up a lot on some of the, most important COVID-related papers, but not enough to mean that we didn't all need the data sooner than journals can do it.
Great. Respondents from the panelists. I know, Susanna, you had talked about metadata and people not publishing papers or submitting preprints, but not publishing the metadata.
Yes, we have done actually last year some work with the COVID Rapid Peer Review Initiative, which brought together several publishers and organization in the scholarly ecosystem. I think obviously with our effort was really more direct to make sure that the data underpinning the publication was there, because the speed and the volume of the preprint meant it was really hard to be able to enforce the type of data policy that you are able to enforce, so let's put it this way, on when you filter and reduce and you manage to peer review the publication. So we definitely realize that even if data is shared, it's never shared in a way that's complete. That's an obvious output, to be honest, something we could have predicted because of the sheer volume of the submission, but also because again it's you need a policy that tell what your author what to do, but you need the tool to allow the author to do what they should do. And we're going to perhaps discuss a little bit the authoring tool and the metadata capturing tool with your next question.
Any other comments from panelists or audience? And otherwise, we'll just segue into the next questions, which Suzanne has already started to discuss. Okay, so the next question is, have automated tools matured enough to assist in the capture of critical information and metadata?
So, well, tools exist. I'm not gonna necessarily delve into if they are mature or not, and I'll try to analyze the problem, give you some insight, at least from my viewpoint. So the tool exists. If we can call them mature is to be seen, really, but also depends whom you ask, depends on the data type. You know, you heard Theo said earlier, obviously for sequencing data where everything is mature, it's a long-term, you know, data sharing has always existed, the norm exists, the tool exists. So depending on the data type, things really vary. Also depends on the maturity of the standards that we need to annotate the metadata to capture these metadata, and depends on the target user. So if these tools are tools for a curator that have familiarity with the information that need to be captured and the standard that need to be used to capture information, then you have a more uptake and you can mature the tool, you can have more impact. If we talk about tools for the data producer, the researcher that have little familiarity with the standard, that becomes more challenging as well. And I want to use a metaphor that Scott Fraser from University of South California used. He said tools are like Brussels sprouts. They are good for you, but not everybody likes them. And actually, it is very true. So if we were to make the standard successful, we have to definitely make the standard invisible. So make the Brussels sprout tastier or hidden between a layer of cheese, if you like cheese, for example. And just very quickly, in terms of challenge, you know, that also affect the maturity of this metadata capturing tool, a lot depends on the readiness of the standard themselves and the level of maturity. And as you know, since Go, Gene Ontology, and Miami in 2001, which are the poster child of the community standard, there are thousands of terminology, model, format, and etc., which have been developed by the community. So this is great, but lots of them tend to be somehow standalone in a sense that they tend to describe a technology or a certain study type. So if you try to describe, or if you develop a tool that try to help the user to capture an experiment that encompass different technology, then you have a little bit of a challenge also for implementation viewpoint, because those standards tend to be standalone and not necessarily interoperable.
Okay, I'm going to backtrack a little bit because we did get some Q&A questions. So from Joe McIntyre, what role could post-publication peer review play in curating papers for better data linkage and metadata capture? So Theo?
Yeah, I mean, hi, Joe. It is, Joe and I were at a meeting that was probably the last in-person meeting anyone held in March of 2 years ago. It feels appropriate to be talking again now. I think the question of peer review is a really interesting one, because some people, as Joe well knows, are adding peer reviews on top of preprints and saying, look here, see how this paper was reviewed. Some journals like the BMJ provide open peer review with publication. The trouble is the peer review isn't really well integrated with the main article. And I find it quite hard to imagine how it could actually deal with the data linkages and metadata of the paper when it is already is one of the bits of poorly linked and badly in need of better metadata standards itself. So maybe I'm missing something there, but I think you could certainly see a world. It is the case already that if authors say the data is available and then it isn't, then post-publication there are opportunities to comment on that and make it available. But other than that, it's hard for me to see how people other than the authors can do that work. Maybe others can explain better technologies for doing that.
Oh, Rodney, I see you had your hand up briefly.
Yeah, this conversation fascinates me because in years of working on standards, I've never been in a room where someone stood up and said, we have too much metadata, right? So this is like in the backlog I carry in my mind of problems that need to be solved. This is when I learned, like my first day at NCBI, and then, you know, how many years later still is not solved. And, you know, I think Susanna made an interesting comment about putting the cheese on the process. And I think how peer review and putting the cheese on the process could work together is something I've noticed over the years. The people generating the data, the data creators, are often doing that for a very specific reason. And they see sort of the things that are associated with their reason for their experiment as important, and the things that are outside that experiment, that reason, as maybe they don't even see them unimportant, they're just not there. But when you talk about an archive that is housing sequence data or clinical data, the archive itself is serving data users, which are taking all this data that's been submitted for these very specific experimental reasons, repackaging it into something that is being used for yet a whole nother set of use cases, right? And so really there is this tug of war between the data submitters and the very specific experiments, and then the archival resources that are trying to package together to serve a whole bunch of other use cases. I think where peer review comes into this is that educational point, where in your paper, you leave out this metadata. And that's really important for the work I do. And when I do this work, I reference your work and take it not only to a new scientific level, but give you credit for that, right? And I think this conversation between the data creators and then the other end users of the data, the bioinformaticians, is a really necessary one to just sort of voice why it's important to have this. And then it's sort of like a recipe, right? Then someone goes, well, maybe I'll try the Brussels sprouts now. Put a little cheese and garlic on it, maybe they'll taste good, right? And I think that's a really critical conversation to have. Anyway.
Great. Andrew, I want to bring you in to see if you have comments about peer review, since you are big on sharing the curation workload and the workload.
Yeah, so I actually didn't, I think Joe's question was fascinating, and I actually wasn't focusing so much on the peer review aspect as opposed to the post-publication, right? What happens post-publication? Because I think there's all sorts of things that can happen post-publication. To Rodney's point, right? I mean, yeah, people publish the metadata and maybe they're not motivated to annotate other parts of their data set, right? I'm thinking now data sets. I'm thinking, suppose, they annotate that I, treated this cell line with this drug at this time point. But somebody else is very interested. Okay, well, I need to know the specific age of the primary cell, the person from which the primary cells were derived, or I need to know a particular factor about the media that you use, right? And so this idea of iterating on the curation, I think, is a powerful one, because if we ask for all the metadata that anybody would ever want, right, we end up with these massive metadata spreadsheets that have 200 columns, and I think compliance with that is really poor, and data quality associated with that is really cool, can be very poor. I see Theo with her hand up, and maybe has thoughts on that.
Okay.
Sure. I just wanted you were reminding me, Andrew, that I sat in a meeting some years ago with David Lippman, who needs no introduction to this audience. And he said, instead of trying to get everyone to curate all their data, when we know that most papers are never cited and never read, let's just do it only for the papers that need it.
Interesting. I wanted to pick up a question from Denise. When it comes to curation of variants and mutations, we rely on tables usually provided in supplementary materials. As far as I know, these tables are not available at the preprint stage. How can we capture this richness of data for our curation then.
So I guess I'll try this one and hope my voice lasts out. Apologies for that. Supplementary files are available with preprints, but they're horrible as they are with journal articles. They're some file that the author has put some stuff in, which is not a great way of curating any data that I know of. So I think that sort of the basics are there if the things you're looking for are supplementary files, but it's not well groomed, I would say.
Yeah, I found it actually very encouraging. Some of the talks earlier were talking about using supplementary material actually in their searches and annotations. So that's great. Okay, maybe we'll kind of segue back to the question on automated tools and the capture of metadata. Here's a comment from Shashi. Journals could incorporate some standards as in journal submission guidelines for reviewers to check. Reviewers would then be required to point out discrepancy in data points included in the manuscript. So anybody want to comment on that? Susanna?
Yeah, like actually most data focused journals do incorporate, I don't know if for standard you mean metadata standard or guideline for reviewer of what to do. So many data journal which data driven or they have data article do have guidelines. The difficulties, as you can imagine, is to ensure that the reviewer actually do follow that or has enough time to do that. And as you know, we are the reviewer, and that's another big issue. So yeah. I think having the guidelines is one thing, make sure that those get executed and executed properly. It's really hard.
Great. So here's another question on the same sort of theme. How can metadata and ontology scale as a function of time as the use cases of tomorrow are not imagined today? And that's a really hard question. Who wants to tackle that?
Well, I can make a comment given I have the microphone on. So it is a race. It's a race that never ends, or it's like changing the wheel of the car as you are moving, because, you need to expand an ontology. There are new terms. You need to have more collaborator, more contributor. So you need to manage the community as well as the content and the technology. It is a real big issue, and it's quite well known. In the past, DNH ran a couple of workshops as well to try to see how to even help funds and promote the work of this grassroots initiative, because let's be very clear, a lot of standards we're using are developed by grassroots initiative, which are volunteer work. And you manage the design, the creation, the evolution, the contribution and the implementation. So it's a hard problem. I don't have the solution. I just sympathize with the problem.
I guess, I would add that I think I try not to be Pollyanna-ish about this, but I'm hoping technology will continue to improve in its ability to, so we used to post PDFs of old manuscripts, older published articles, and people complained, but they're just like a Xerox copy. But these days, optical character recognition can, extract much of the text and make it more useful. And in the same way, we're getting better at extracting sentiment and meaning out of text. I got to hope that we'll be better at extracting the key metadata that we decide in future is useful.
If I can make a counter comment what you are saying, yes, there are technology where you can enrich ontology using text mining. The thing is, you have to build ontology that are useful for the specific use case. So you always have this validation phase in which are we meeting, is the ontology answering the question that the researcher wants to, or the key metadata in the field. So I'm not sure the type of validation evaluation steps it can really be automatized, or at least it's been very hard with the group I work with. And I see there, Rodney has a hands up.
So I love conceptually ontologies, but I think you hit on a really major point. They're typically developed by specific scientific communities that are interested in specific types of information. And it's very hard to build sort of the ontology of everything, right? And I think also the concept of an ontology is a little bit different than a concept of normalized terms, right? And so one could have, and I see Andrew making a face. So what I mean by that, an ontology typically represents a hierarchy, right? And the hierarchy being important for sort of the understanding of how a term fits in with other terms. Whereas there's another sort of problem, which is I have disparate terms, or I have terms that are reused in inefficient ways, and that's just normalizing, in my mind, the vocabulary, like having a normalized vocabulary one you can use, and then having that available to be parsed and placed into ontologies. That seems like two different steps to me. And I think that just having a conversation that everyone can understand about normalized vocabularies is really critical to informing users, both in terms of data generators and people working with the data, that this is an important concept. Include the metadata, include these sort of normalized terms, and then when this stuff is available in your scientific community, people can come along, parse that, and build it back into an ontology. And I actually have faced this problem a few times. One of the problems, we've always wanted to map our stuff to ontologies. I'll just be completely honest about that. Every time we see a problem, we're like, who else is solving this so we don't have to solve it? But when we ever engage groups around ontologies, the two issues. One is the fact that different ontology groups are trying to solve different problems for different scientific reasons. And then two is speed. We need to process 5 million records today. We are going to discover 100,000 unique terms. And then the ontologists are saying, okay, submit those and we'll review those over the next 2 or 3 years and integrate them into our ontology. And it's not to say that's a bad process. That's probably a very good process, but it's not a process that serves sort of rapid change, or a process that serves sort of that first line, that first draft of making the data interoperable.
Yeah, that's a great point. Andrew, did you want to comment on this, or?
I would only say that I think focusing on process is exactly the issue, and I think that would, the speed with which ontologies can innovate and adapt varies widely depending on the ontology communities, because as Susanna pointed out, a lot of these are just sort of volunteer efforts, and so there's huge variability. So I would just say that, to me, I would guess that, whatever 2 years would be on the long end of things. I would imagine that there are also, very active ontology communities that I think would be and have been much more responsive, in this time of pandemic.
Yeah, I was trying no flack at ontologies. I love the concept of ontologies, and I think it's a critical concept going forward. But I was actually just trying to make the point there, if you're trying to work rapidly, that may not be your first line of metadata normalization. But retrospectively, I absolutely agree.
And I would just say that there are certainly ontology communities that are equipped to respond in real time. Yeah.
Yes, a selected number, I would say.
Okay, we unfortunately lost some very interesting questions out of the Q&A.
They are in the answer. Solinet, I realized that as we answer, they disappear from the open. So sorry, I realized it too.
Okay. Yeah, I'm not going to try to do this real time, but anyway, there's some very interesting questions that got answered. I'm just reading these. Okay, a comment from Amanda Hicks. As an ontologist, I agree with everything Rodney said. Is there some way that implementing normalized terminologies can give us empirical data to help develop more ontologies more rapidly, especially for consensus-driven ontologies? Yeah, seems like a very practical suggestion.
I was going to just say something on that. along those lines. One of sort of thematically things that we've been working with at NCBI is the idea of making the problem obvious to stakeholders. And we see sort of our movement to the cloud service providers, these open access cloud platforms that don't cost money for people to work on, as a way of doing that. And what I mean by that is if you have some processes running internally and they are giving you sort of these normalized terms, you can put them out there and see how the rest of the stakeholders can use these normalized terms. If you are encountering data that's missing, the metadata just is absent in those fields, you can see that, the people can actually see whether it's there or not there at a very large scale and kind of do those and get those metrics. And how often do we see these terms submitted? How often are they submitted and ambiguously or without, sort of defined vocabularies, et cetera. I think that there's a real opportunity here for sort of building on these themes and just throwing the information out there in ways that people can work with it at scale, that people can unleash the machine learning algorithms and unleash the other approaches to trying to really put metrics around the problems. And then that can feed back into where we need more work, right? Just something we've been working with over the last couple of years as we've moved data to the cloud, because the cloud allows you to put a lot of information in place in a way that I don't think is easily done through web resources.
Yeah, I think that's a great segue into the third question. We may have already touched on this a lot. Could curation be scaled by sharing the workload more broadly? And I think some of these approaches are addressing parts of that.
Yeah, if I could chime in there, because I'm very interested in obviously shared effort. And that to me has been one of the most notable things about pandemic is that, right, everybody, right, there's been such a groundswell of interest in sort of trying to help organize information, right? So the thing that sticks out in my mind was actually the COVID tracking project. I don't know if, this is probably distant memory now, but right, in the first year to year and a half, right, when, truthfully, I think what happened is that, volunteers sort of, came into a space where they saw a gap, right, in terms of tracking epidemiological data on a local level, right? And so this started out with a shared Google Doc, I think, right? And then, people were gradually organizing structure around that, and it filled a vacuum that existed for a variety of reasons. But then, right, COVID tracking, I think it was a year and a half into it, said, okay, now that CDC has got data together and is reporting that well, great, we can retire ourselves. But the speed and agility with which, and perhaps standards free or loose standardization, help that effort. But the ability for people to organize there, to me was pretty outstanding and is indicative of sort of similar efforts elsewhere in this pandemic as well.
Okay, Rodney, do you have your hand up or is that left over?
It's my hand up. Sorry to talk so much. You're getting your money's worth out of me today. So yeah. I would take what Andrew said and take the question and kind of turn it on end a little bit. I totally agree with Andrew, and that was a great effort. And I think there are two key components here. No one has the funding to solve all the problems. It's just end of story, can't be done. So the question really should be, how do we do it? Because we have to do it, because there's no other solution, right? And then Andrew's point is really well taken, because we've seen this in a number of different resources that have been developed over the last few years, including NextStrain, and including outbreak.info, where academic groups that are not constrained by all the previous promises they've made and all the ongoing work they have to do can move very quickly to solve problems, right? And you saw that at the sequencing level. A lot of the data producers during the Delta and Omicron waves, a lot of that data surge was led by academic data creators. And so I think we need to sort of take all the component parts and make them work together really efficiently, right? And that is, who are the sort of emergent solution people, and who are the folks that can come along and look at those emergent solutions and build sort of hardened production ready ways of serving those same use cases, right? And start seeing the community less as a bunch of competitors and more of a group of folks that are exchanging handshakes to make the world a better place, right? Because that's what we're all after, right? And that gets into this whole idea of how can we all work collaboratively and each of us get sort of the credit we need to keep our jobs, to keep our grants, to keep our institutions going, right? But I think it's sort of critical to integrate these pieces.
Yeah, Susanna.
Yeah, I can add also my, I won't say experience, because we are trialing now the idea of creating community curation around our fair sharing resource, which is data in a sense of data about standards, data about repository, data about policy. And so we are trialing this idea of curation as a team sport, because curation is a team sport, but we also want to see how we manage to activate the community, how much interest there is, what social code we need to put in place to make everybody not just welcome but credited, and that's very important. I also see that Lindsay Anderson, which is one of our, first actually community curator, it's also on the call and asking questions. So we will test and trial if this is working, if like I said, the community gets something in return. And this can be somebody who has expertise in curation or somebody who wants to learn what curation is, because we will try to give something back as we gain their expertise, try also to train perhaps the new people to appreciate and understand what curation task means. And again, curation can be a career job, hopefully.
Yeah, I was very interested that in Erin Riggs' talk, she talked about volunteers and training them and so on, which is a really interesting concept. It long-term, it may scale. Short-term, it probably doesn't, it can't happen at the speed that we need it to happen sometimes. I'm going to take this lull in the conversation to read Anita DeWard's very interesting comment. So Anita said, I don't think we can identify which papers need curation a priori and remain inclusive and open to to work confirming or not prior results, which is important for reproducibility. In fact, for those papers, data curation would be more important since they might not be scrutinized as much by readers. Yeah, Theo apparently answered that she doesn't think it's a good idea. So I don't know, Theo, do you want to?
So I think Anita was, I related what David Littman had said, that we should only curate those data sets that prove to have a value to people. And Anita was saying that's not a good idea because we don't know a priori which ones we're going to need. And I was agreeing with her that I wasn't saying it was a good idea by reporting it. I was simply saying that the problem with, with not doing it all up front. I mean, I think I didn't say this, but when you talk to the authors of papers and you require data availability, for example, they've already lost half of what they needed at the time they've written a paper and are submitting it, let alone if you went back to them, after it was published and at the time that someone realized they want something, the postdocs left the lab, the data's on an old laptop, etc. So, I mean, I'm definitely actually a fan of saying we do it as much as possible at the time that, ideally at the time the work is done, but certainly as soon as it's complete and ready for sharing in some way, then that's the time to share the data as well.
Okay, thank you. And another couple comments that I'll read. One from Linda: the more requirements that we make for database submission, the more submitters complain that we are making it more difficult. How do we balance metadata capture while not discouraging submission? I think that's a really key question. So who wants to take that up? I saw somebody's hand briefly, but Rodney.
Yeah, I think I addressed it. Hey, Linda, I think I addressed this before. I think this is always the balancing act, right? And to go in a little bit more detail, oftentimes, not only is the data creator sort of not aware of how the data is going to be used one day, sometimes the data creator is removed from the data itself. So there is a group that collects samples. There is a core facility that takes those samples and turns them into DNA sequence. There's a bioinformatics core that then assembles those sequences and puts them in the context of all the other data they're collecting. And then there's some group that's submitting. And these can all be disparate. And along the way, metadata can get lost, and it can be very difficult to go back and try to reconstruct the lost metadata. And we've encountered that over and over again. And again, I think it's education, right? It's true. If you build a high wall, few people will have the energy to jump over it. It's really that simple. We all get that. And you're always playing a balancing act. But if you make folks aware of how their data is being used long-term, then things get sort of visible. They get real for the data creators. So if, and then, there are lots of ways of doing this. You can have, sets of data that are sort of your gold standard sets of data. And those include, 10 metadata fields. And so when someone goes and select those and they don't see their own data, they go, why is my data here? Well, because you didn't enter all these metadata, right? Or there are other ways where you have, graphical interfaces, like some of the ones that Andrew mentioned earlier, that really draw on the metadata to allow people to visualize things. And then again, people will notice that their data is not part of the visualizations. Why am I left out here? Because, well, you didn't provide the metadata, and we're not using your data in this visualization because the metadata is not there. And then you can also, make it widely available in places like the cloud, and folks can do the analytics and come back and talk to submitters directly. We see you've got a lot of great submissions here. The DNA sequence quality is really great, but you're missing all these terms, and those are really important. And it's just a matter of talking to one another over time, and it's not going to happen immediately, right? People have to see the value in their activities before they're going to spend time and energy in those activities.
And I think we have segued actually nicely into the last question. How can standards keep pace when new knowledge is rapidly emerging? Which is really one of the things we've been very much talking about. I will try to read a long comment from Lindsay. I'm going to skip some of it, but it's an interesting comment. There are gaps in machine actionable sets of normalized terms or controlled vocabularies for defining the information objects that are contained in a schematic knowledge framework versus the ontologies contained in an informed structured metadata file for linkage to multivariate data sources in a graph-based setting. There's a cross-linguistic crosswalking effort still needed for extending metadata structure to include both merging universal semantic object terms with structured data, structured metadata ontologies. So I think that's kind of what we've been talking about here. Anybody want to comment on this or follow up with something? Okay, we will go on to another comment from Joe. If we as a community put as much effort into good metadata creation as we did trying to get papers published in a small number of journals, we might get somewhere. How to shift this needle? Unfortunately, I think part of the question is funding and credit assignment.
Exactly. So the real the reason people put so much energy into where they publish is that they believe, rightly or wrongly, that their institution and their funder judge them according to where they publish their papers. If they believe that their institution or their funders judge them by how good their metadata are and whether they deposit data with their studies, then they'll start doing that. And certainly some funders are trying to do that. And of course, NIH moving in that direction as well. But even Wellcome, which had a, notably had an open access policy that found, only 70% compliance, even when it had enforcement officers. I mean, it's very difficult for anyone to create the sticks. Someone else in the comments said, we've tried a lot of carrots. Is it time to try a stick with people who don't want to eat their sprouts? That to mix metaphors. I think it's all very well, but the incentives are simply not there, and we have to find ways to create those incentives.
And this is, I think there's a related comment here. What is the role of the author, end user, publisher, funder, and repository in the context of this discussion? How to harmonize these groups in terms of fixing the issues? And I think that does raise the question, doesn't answer it. Andrew.
I'll just say, since we have lots of NIH folks on the call, I 100% agree with Theo's comment that NIH to some extent could wield more sticks. And I think, sorry, the data sharing requirements that are going into play early next year, right, are part of, pushes along that regard. So I'm not saying that there's a lack of appreciation or action on this, but in general, I do, to take just the open access requirement, right, PubMed Central, right, is a great example of when NIH wielded a stick, and I think that resulted in a fantastic resource.
Yeah, I don't know if there's anybody from the NIH side other than Rodney who would like to respond to any of that.
I'll quickly say that certainly I've heard that comment many times before.
Yeah, this is Kim speaking, also from NCBI. I think NIH tries to walk a line between providing incentives and supportive policies to help with data sharing and help with submitters, and not providing something that turns out to be an undue burden on the research community. And I know that NIH takes both of those into consideration.
And this is Mike Herter from the National Library of Medicine. And I'll say that I think, as we'll hear Susan Gregorek address, NIH is doing a lot in the non-policy area, but we are also doing a lot in the policy area. And there are limits to the kinds of things that NIH can actually do. But we are certainly cognizant of the things we can do. And we are working closely with partners in academia and elsewhere to move these things forward. So this is really a point of lots of discussion across all of NIH, but it's a good point, Andrew. Thanks.
All right. Any further comments from any of the panelists? And if not, oops, I got another couple comments. So Heidi says, I would like to see creative socio-technical solutions where incentives, sticks, policy, and technical are integrated. Yeah, that would be excellent, and good to hear that NIH funders are certainly thinking about this.
And by the way, just to make the obvious comment, that would sort of be the attributes of an integrated scientific community.
And one other comment from Lindsay, who wants to follow up. There are a lot of scientific community, external community working groups doing amazing work on these issues, which is important to stay relevant on those global developments. So yes, there's a whole lot of activity. And yeah, one of the things I was struck with, especially on the metadata side, was how many different metadata sort of standards there are for many different things. And I mean, we're going to pretty soon need a directory of metadata standards to keep up with this. So yeah, there's a whole lot of activity, and how can we best funnel it and enable curation at scale, and the enormous energy that the community has shown at both the scientific community and the community at large. So I'm going to conclude the session. Thank you very much to the panelists and to the audience for a great discussion. And I think we have a 10-minute break now.